Good morning, all. Happy Friday. Uh, the weekend's coming up. So I looked at the calendar and it turns out that we only have 5 weeks left of class. So 4 weeks in a row, and then a spring break and the last week of class. So not long left till the end of the semester. I hope you're excited about it. I hope the spring break, not the spring break, but the summer break appears exciting to you. And especially with the rollout of the vaccines, it looks like social distancing is almost over and lack of social gathering is almost over. So I hope you're as excited about it as I am because it's been too long. All right, are there any questions before I start the lecture? All right. So, few reminders, as usual. Quiz 7 is due today. So if you haven't taken it already, please make sure to take it by 11:59 p.m. today. Quiz 8 opens tomorrow, and it is going to be due on next Friday, so in a week. And we have a participation assignments that is due today. There are 3 questions, quiz as usual. So please make sure to complete it by the end of the day today. So I'm going to, last time I covered the evidence of evolution. We've looked at the relative dating, absolute dating. We've looked at the homologous structures, anatomical structures versus analogous anatomical structures. So today I'm just going to continue with the evidence of evolution lecture and go into, probably this lecture will spill over into just going over evolution, forces of evolutionary change. So another source of evolutionary clues is comparing embryonic development patterns across species. So in this picture shows the similarities between skulls of the chimpanzee and human fetuses, and the dissimilarities of the skulls of the chimpanzee and a human. So when at the fetal stage, whenever a chimp and a human are fetuses, they look very similar. But once the fetus is born, and gradually develops into adult, we see more and more dissimilarities in the anatomical structures of chimpanzees and humans. And this is because the kind of proteins that are expressed, the kinds of genes that are expressed, that they differ between chimpanzee and humans. But overall, the genotype is very similar between chimpanzee and humans, and partly that is why the fetuses look very, very similar. So in fact, many vertebrate embryos appear very much alike. So in this picture, there are 3 embryos. One embryo is of a chick, another one is of a mouse, and a third embryo is of a human. So note that all of the embryos, the chick, mouse, and human, they have tails as well as other similar homologous structures. They all have like common features, but as an embryo develops into a baby chick or baby mouse or baby human, suddenly these babies don't look similar at all. But it is quite interesting, and I find it quite fascinating that at embryonic stage, we all very much look alike. So what influences this development, these differing features of the bodies once after the embryo develops into a baby mouse or baby chick or a baby human? There are specific kinds of genes that control the embryonic development, and these genes are called homeotic genes. So they are needed for body structures to develop normally, whatever normal is for a given species. So small differences in these gene expressions might make the difference between, for instance, a limbed and a limbless organism. Let's say we take an embryo, look at the embryo of a chick and the embryo of a python, okay? And let's say we have two kinds of homeotic genes in there. So homeotic gene A and homeotic gene B. Homeotic gene A is noted in blue and homeotic gene B is noted in green. So in a chick, we see that some portion of the body of the embryo is going to have the tissue in the some portion of the body will have the homeotic gene A expressed, and some portion of the body will have homeotic gene B expressed. In an embryo of a python, let's say we have the same genes, but both of them are expressed in the same body parts. So the cool thing about this hypothetic, well, not hypothetic, but abstracted, I would say, gene A and B, is that if only one of them is expressed, the limbs develop. But if both of them are expressed at the same time, none of the limb develop. So whenever the gene A in blue is expressed and gene B is not expressed, the forelimbs develop, like the upper limbs develop. And whenever gene B is expressed and gene A is not expressed, then the hind limbs develop. So in a chick, there are regions in the fetal tissue that express only gene B, and there are regions there is a region in a fetal tissue that expresses only gene A. So the chick will have both forelimbs and hind limbs. Whereas in a python, the tissues along the embryo, they express both gene A and gene B, and therefore no limbs are going to be present in a developed python. So, any questions about the homeotic genes so far, which allow which promote body development, which facilitates the development of the structures of our bodies?
So, could there be a chance of like a gene alteration or just some weird thing in one specific thing that like a python could have legs that don't do anything?
Uh, yeah, it potentially it could happen if there is some sort of mutation or like um, some random thing that happens during DNA replication, and the fetus will have maybe uh, the fetal tissue will have forelimbs expressed in a small region. So a python may have like some python may have forelimbs. Probably will not survive, but yeah. Um, but mostly the pythons will have the will not have the limbs because both gene A and gene B are going to be expressed. In fact, sometimes I'm not sure if you guys remember, but many lectures ago, I showed a picture of a fly that had legs in the place where, like in the mouth. Okay, the legs grew from the mouth, and that was caused by a genetic mutation, and in fact, that was caused by a genetic mutation in homeotic genes. So whenever the homeotic genes kind of go crazy, uh, the hind limb homeotic gene can be expressed where a homeotic gene A should be expressed, the one that regulates the forelimb. Okay, so all kinds of weird things can happen with the homeotic genes, and these weird things, these weird mutations, can manifest in very deformed individual, can result in a quite deformed individual. Doesn't happen often, but sometimes it does. And especially it's visible in species that are so-called R strategies that have lots of babies in a small amount of time. So like flies, they reproduce with astronomical speeds. So in flies, there's more probability to see some freaky mutations. Like 1 out of 1 million flies will have some pretty freaky structure. Any other questions? Okay. So since 1970s, biologists have compiled a wealth of data by comparing molecules inside of the cells of diverse organisms, particularly by comparing DNA of various organisms. And in general, the whenever the sequences of the nucleotides within a DNA are similar between two individuals or two species, that gives a clue about the common ancestor of the species, because the sequence similarities are inherited from the common ancestors. It is highly unlikely that two unrelated species would evolve precisely the same DNA, because DNA contains a lot of nucleotides, all right? It contains millions and millions of the nucleotides. And imagine like the mutations are random, okay? And if the random mutations of millions of the nucleotides in the millions of the locus loci, it's highly unlikely that these millions of mutations will be repeated in two unrelated species. So if the sequences of the nucleotides are highly similar between the two species, that means that these two species have a common ancestor. And what it looks, biologists frequently depict these similarities in the form of the phylogenetic trees that we already looked at. So these phylogenetic trees, they are basically derived from the similarities in the sequences of the nucleotides in a species genome. So we can approximately estimate, and this is really cool, but we can approximately estimate the timing of the divergence between two species by comparing the dissimilarities in their DNA. So we already covered like kind of two dating techniques. First technique for dating was the relative dating, and it dealt with us kind of digging a hole and seeing, and the deeper we went, the older the finding would be. Whatever remains we find deeper in the ground, the older they will be. But the second dating technique was absolute dating technique, and it had to, we looked at the carbon dating, but apart from carbon dating, there are some other dating techniques like the strontium dating and lead dating. Virtually like any isotope that you have, you can date things with any isotope, using any isotope. Carbon dating is pretty common, dating things that are less than 100,000 uh, years old. So the third type of dating is the so-called molecular clock. The stable mutations in the DNA, the mutations that are inherited over time, uh, are pretty regular. They occur like once in 25 millions of years. So in this picture shows the divergence of lemurs. So the two species like of lemurs have lots of common DNA chunks, but they have some dissimilarities in the sequences of nucleotides. But a lot of portions of the DNA between this species and this species is similar. So these two, because the lots of the nucleotide sequences are similar between two species, they had a common ancestor. And let's say this is the common ancestor of these two guys. And as the time went on, and let's say nothing influenced there was no selection for a particular phenotype or genotype. Let's say there was a random mutation in one nucleotide, and this random mutation happened after 25 million of millions of years, and after another 25 millions of years, there was another mutation in the genome of the common ancestor. and that led to species A, and two other mutations over 50 millions of years led to the species B. So the mutations that become stable in a population, they are pretty regular and occur at some steady frequency, like usually once in 25 millions of years. Um, what else do I want to say? And it's important to note here that I'm talking about the kinds of mutations that are not selected for. And it is my understanding that this situation is pretty rare, because usually the whatever mutation, if a mutation does not increase the probability of survival of a population, of a species, then the individual with that mutation has a higher probability of death. So that mutation is not conserved in a species over time. So although it is possible to date a species, the age of the species over time with using the molecular clock, it's it's pretty tricky to control for the natural selection. It's pretty tricky to prove that this particular mutation did not benefit the species, that it was not selected for over time.
Are these just a kind of off track question? Is this done all through like statistical odds of like on the professional side, like odds of getting
Yes.
mutation? And wow, yeah.
Yeah, exactly right.
I think out of all the ACE and AP classes I took, statistic was the only one I didn't pass.
Statistic was the only one that what?
That I didn't pass.
Oh no. My condolences. Well, I hope you find a good professor or a tutor
Yeah.
in statistics, because it's super interesting. It can be super interesting and super useful. I mean, if any of you are planning to go into like data science in any shape or form, data science is all about statistics and all about regressions and all about, like, even the complex machine learning techniques. They are rooted in the very basic statistics. Like, if you get the basic stats, if you get basic regression, basic like least squares estimation, you're good with machine learning. You pretty much can learn anything else. So calculus and stats, very useful for data science and machine learning, if your career path will take you there, or if you decide to change it and become a data scientist. All right. Any questions about molecular clock, homeotic genes, anything I've covered so far? Because the next I'll go into forces of evolutionary change.
I have a quick question.
Go ahead, Madeline.
So you said that this random DNA mutation from the lemur wasn't naturally selected, but then you also said that it's hard to prove whether it was or not. So why do you think an entire new species emerged if it wasn't helpful?
So in some environments, it may not be helpful. The random species may emerge randomly. that's exactly my point. So if the particular mutation is not selected for, it's the species may still come to be just because this mutation occurred. I'm kind of garbling things. So
Could you compare it to birds of paradise? Like there's a million birds of paradise. Are those mutations really necessary? Well, you could argue no, that they're not super necessary. I mean, they're more for mating, and yet
What's an
we
example of
have.
that kind of mutation though?
Oh, if you Google birds of paradise, they have all different types of dances and shapes and colors, and you could argue that none of those, the dances that they learn or the shapes that they can make with their bodies aren't necessary for survival, yet that they have happened on this island because there's a plentiful amount of resources. So it doesn't matter that one bird can turn sideways and go orange, and the other one can turn sideways and go blue. There's enough food for all of those birds of all the variety to live. regardless of their pointless mutations.
Yeah, that makes sense. So a random DNA mutation is not necessarily helpful, but it's also not harmful at all either, because obviously that species didn't die.
Yeah, so like say like you were somewhere cold and there's a random mutation that one of the lemurs, he has a thicker coat than the rest of them, right? And there's one season where it's really cold and that mutation just happened to help out, and all the rest of the lemurs that didn't have that longer fur, they die. It's all really controlled randomness. It's not, none of it's planned, obviously.
Thank you,
Okay,
Dylan.
all right. Thank you, guys.
Yeah,
That was an excellent example. Thank you, Dylan.
you're welcome.
All right. Does anybody want to discuss anything else related to molecular clock, homeotic genes? Okay. So next, forces of evolutionary change. First, the definition of evolution. Evolution is the genetic change over time. So evolution occurs when there are changes in heritable traits from generation to generation, and some changes lead to the development of different species. So if evolution occurred, if the population has evolved, it doesn't necessarily lead to emergence of a different species. So populations can be pretty different, and even individuals within the population can have pretty different phenotypes. So an example of a pretty different phenotypes within different populations within one species is basically, we can look at us. We have very different phenotypes, but we are still the same species, Homo sapiens. But there are tall and blonde people, and there are people with dark hair. And all, like originally, it's the people were separate, like the populations were separated, and the traits such as light skin and light hair color were beneficial for survival in or rather were selected for in like northern regions, whereas dark hair and almond shaped eyes kind of were beneficial for survival in some other regions. So again, the species is the same, Homo sapiens, but we have different phenotypes. So yeah, that is kind of an illustration that evolution may lead to different phenotypes, but may not lead necessarily to divergence of species, to emergence of different species. So, what is important to understand about evolution? One of the things that is basic things that is important to understand about evolution, it does not occur in individuals. Okay, since the individual's alleles do not change, it occurs in populations. So in many individuals living in the same kind of regions. Um, again, and the reason for evolution not occurring in the individuals is because individuals' alleles do not change. We inherit the alleles from the parents, and kind of that's it. We're stuck with them. But in the populations, allele frequencies change from one generation to the next over time. And some alleles become more common in the populations, and other alleles become less common. So like in population in the Nordic countries, the alleles for light hair, blue eyes, and quite tall height, ugh, I'm jealous, become very frequent, became frequent, and in populations in, let's say, Southeast Asia, the alleles for dark hair and almond-shaped eyes have become more frequent. So these differences indicate basically that the gene pools in Nordic populations and Southeast Asian populations are different. But both populations have common ancestor, but their genotypes have diverged, likely due to the effect of the environment over thousands of years. Okay. So any question about this notion that evolution, is this pretty clear that evolution does not occur in the individuals? Like, and any one individual cannot evolve, populations evolve over time, because we are looking not at the individual's alleles, but allele frequencies within the population when we're studying evolution. We good with that? All right. So if we're talking about evolution, of course, I cannot not talk about Darwin, Charles Darwin, who was a Cambridge-educated British naturalist, and he's famous for publishing the book on the origin of species. So Darwin, as a naturalist, he led a pretty good life. I mean, when I read about his biography, it was pretty sweet. I mean, he was this guy who bred like who was very into breeding dogs and pigeons, and he's basically was the job of a naturalist was literally to travel around and describe species and kind of think how these species came to be and think about lots of other things. So one of his expeditions was a 5-year journey on Her Majesty's ship Beagle, and his role on the ship was to be a collector and captain's companion. So apart from collecting species when they were at sea, which was, I imagine, most of the time, he would kind of, I would imagine, drink with the captain and have a good time. So he described species once they came on shore. He described species and noted individual, and he noted that individuals differed within the population. Not any two individuals within the population were alike. So this was his kind of first observations as he traveled around the world and studied the species and populations of the species. He noted that not all, not any two individuals are exactly alike. Like not any two individuals are basically identical twins. And he was pretty intrigued by that. But most notable findings of his were made in Galapagos Islands, which are right here. So he described 13 distinct species of birds that seem to specialize on particular seeds or insects. So he basically collected these birds, and he described like that the beaks were different and the shapes of the heads were different. And eventually he asked his buddy to identify the birds, and his buddy said, well, those are all finches, buddy. And Darwin's like, no, well, they're different species. Like, look at how different they are. But basically, that allowed him to start getting some ideas about evolution, about natural selection. So Galapagos Islands are a pretty cool place to study evolution because they're the kind of food that would be available to birds differed, and they are separated by sea. Okay, so the migration between the islands is not that great. So the Darwin hypothesized that the beaks of the different species of finches, they kind of came to be because of the kind of food that was available. So the species with larger beaks, I mean, the individuals that had larger beaks survived because they could eat the food, the kind of larger seeds that was available to them. And individuals that had the food that had smaller beaks, they survived because the kind of food that was available to them was small, like kind of fit the beak. So then Darwin proposed, after thinking about it for a while, Darwin proposed this idea of descent with the modification and natural selection. Or basically that idea can be summarized as best suited individuals would survive and reproduce. So the idea was kind of on the surface, because being a breeder himself, he was very familiar. Darwin was very familiar with the idea of artificial selection. So artificial selection has been going on for ages, particularly in the agriculture. For instance, the broccoli, the Brussels sprouts, the cauliflower, kale, and cabbage All of those guys were artificially selected from wild mustard plants. So well, not we, people before us, they selected for various traits of the wild mustard plants in order to get all of these delicious vegetables that we have now. So uh, remember that no two individuals within a species or within a population are exactly alike. So some wild mustard plants would have, let's say, more pronounced stems and flowers. So people over generations of a wild mustard plant, if you were to select only those individuals within a population that had pronounced stems and flowers, over, let's say, 100 generations of plants, you would get broccoli. If you were to select only individuals of wild mustard population that produced like lateral buds, over hundreds of generations, you would get Brussels sprouts. If you would select the individuals that from the wild mustard plant that pronounce the flower clusters over 100 generations, like flower clusters right here, um, over hundreds of generations, you would have cauliflower, and so on. So, and by selecting the individuals, I mean selecting the individuals and allowing them to reproduce further, and further selecting the individuals with traits that are desirable to you. So it's pretty wild, at least to me, to realize that all of these different looking vegetables were selected from a wild mustard plant. Okay, and same for dogs. I mean, we have a crazy variety of traits in dogs now. We have crazy amount of breeds, and all of those breeds were virtually generated from like wolf. And so various breeds, like as examples, are Cocker Spaniel, Collie, Dalmatian, and German Shepherd. I have a boxer who was selected up to Yin Yang. Um, so these are all examples of the artificial selection. Okay, and for Darwin, it was kind of a natural jump to go from artificial selection to natural selection, where nothing like the the selection of individuals within a population occurred just by the virtue of being able to to obtain food, or being able to reproduce more successfully, and so on. So, what else I wanted to mention about artificial selection is that unfortunately, I'm going to go back to the dog breeds for a bit. Unfortunately, with the dog breeds, artificial selection often introduces some undesirable traits. So let's say we have the dogs that are small and cute, and we kind of selected for these traits, but also these dogs are often sick, or they have higher probability of developing some genetic disease. So a couple of examples of the genetic disease, like German Shepherd is very prone to have hip dysplasia, or Great Dane is very prone to have heart failure and bone cancer, and Labrador Retriever is prone to having dwarfism and blindness. So all of these are drawbacks of natural selection. And this is because the traits that we select for, the genes that encode the traits that we select for, are located on the same chromosome that the undesirable traits, such as deafness, blindness, dwarfism, and so on. Okay, so because we cannot physically separate, the genes on the same chromosome, if this one gene will encode some cute feature, and another gene on the same chromosome will encode some disease like deafness, then we will have a cute but deaf dog. Okay, well, a cute dog that is also has a higher odds of being deaf, and so on. So does it make sense, like uh, why some desirable traits are often associated with genetic diseases? Are there any questions about that? Okay, so just to reiterate, the desired features on the phenotype, the genes that encode the desired features are often located on the same chromosome as some other undesirable traits, such as deafness or hip dysplasia, and so on. And artificial selection for the desired features cannot different like cannot exclude the undesired features due to both genes located on the same chromosome. All right, so back to Darwin. Darwin proposed natural selection as a mechanism for evolution. So natural selection was kind of made sense to him because he was very familiar with artificial selection, and he's seen a lot of populations and a lot of individuals within the populations with across a lot of different species around the world. Okay, so what he observed, he made basically three observations that led him to the idea of the natural selection. The first observation was the genetic variation. Within a species, no two individuals are exactly alike, and some of this variation is heritable. So the large beaks in finches are heritable. The baby birds will have large beaks like their parents. The second observation was that the resources were limited. Every habitat contains limited supplies of the resources required for survival. It could be food, it could be like physical space, it can be water. And third observation that he made was that species produced much more offspring, many more babies than were sustainable. Okay, so more individuals are born than survive to reproduce. A lot of individuals die and do not get to reproduce. So what he made three inferences from his observations. The first inference was that individuals compete for the limited resources that enable them to survive. And this inference was heavily influenced by the work of his contemporary, Thomas Malthus. And Thomas Malthus was a British economist. Well, he was more than that, but he wrote a lot of basically works on the economics, and that's basically his kind of his idea in the economics that the resources are limited and we kind of compete for these resources. So individuals compete for the limited resources that enable them to survive. It's true in economics and it's true in nature. The second inference that Darwin made was that there was an unequal reproductive success or natural selection. The inherited characteristics of some individuals make them more likely to obtain resources, survive, and reproduce. And finally, the third inference that he made based on his observations was the one of descent with modification. So, over many generations, a population's characteristics can change by natural selection, even giving rise to new species if the differences in the genotype become sufficiently large, sufficiently substantial. So, evolutionary theory is often misunderstood, and here this table shows the three common misconceptions associated with the evolution. So, the first misconception says that biological evolution explains the origin of life. This is not true. Then this is a misconception, okay? Biological evolution did not begin until life existed. So by definition, it cannot explain the origin of life. It only can explain how life evolved once it's originated. The second misconception is that evolution is a random process. Mm, it's not quite true because the environment kind of selects for the species that are the individuals that will survive. Okay, so some mechanisms of evolution, such as mutations, do occur randomly, but natural selection is non-random. because the environment selects against poorly adapted individuals. Whoever does not survive in a given environment will die, and therefore will not give their genes to the next generation, will not transfer their genes to the next generation. Somebody unmuted. Did somebody want to ask a question?
If anybody even remembered, I said earlier, imagine you have two sets of animals. One randomly has a longer haired coat than any other. And then, recently we just saw in Texas, cold storm, like that happens in nature, the random event, colder than usual. You have another random mutation in your monkey, have longer hair. He stands a better chance of surviving. And both the events of his long hair and that cold storm were two random events.
That is an excellent example. And in fact, you also touch on the concept of importance of diversity within a population, of genetic diversity within a population. And we will discuss it, I think, during next lecture, the lecture afterwards. But this genetic diversity is very important exactly for the reasons that Dylan touched upon. Like if there is an extreme event that wipes out most of the individuals within a population, if all of the individuals are genetically similar, then the population will be wiped out, and maybe the whole species will be wiped out if all of the individuals within a species are similar. But if they are diverse, there's a higher probability that that diversity in phenotypes, at least one, will facilitate survival of the extreme event. So another, I'm not gonna give that example. I'll save it for later. But thank you, Dylan, for chiming in. And it's a great example. So the third misconception is that in a changing environment, all individuals in a population simultaneously develop beneficial adaptation. Again, not true, and that kind of goes back to the second misconception here. The adaptations become fixed in a population over multiple generations, as individuals with beneficial adaptations are more likely to survive and reproduce and pass their genes to the next generation correspondingly. Okay, so not any one individual cannot evolve. Only the population can evolve again, because whenever we inspect the population, we look at the frequency of the allele within the population gene pool. All right, is there anything else I wanted to mention here? I don't think so. Any questions so far? Okay. So natural selection molds evolution. Natural selection does not create alleles, rather it strongly selects for alleles that arise by chance. So in the picture here, you see a seahorse, which is pretty hard to distinguish from the coral in the background. And that is because over many generations of seahorses, the ones that had this phenotype survived, because for the predator, it was hard to distinguish a seahorse from a coral. But all other seahorses, maybe that have different color, maybe that who are bigger in size or had various other potential phenotypes, they eventually died out and did not get to reproduce and pass on their genes because they were eaten by predators. So natural selection requires genetic variation. It does not create alleles. Okay, if all individuals are identical, change in environment wipes out the whole species. So as Dylan mentioned before, so genetic variation is required for the species to survive. And we kind of discussed all of them. Other things. So some other things that I wanted to reiterate is that if all the individuals within a population have the same genotype and the same phenotype, or very little variation in genotype and phenotype, that some catastrophic event such as deep freeze or disease outbreak or what have you has a higher probability of wiping out the whole species. But if a species has a high diversity in the phenotypes, then a deep freeze or an outbreak of like high predator population or disease outbreak have higher, the species has higher probability to survive. There's higher probability that at least like a couple of individuals within a species will survive whatever extreme event will come their way. So reproductive success is enhanced by adaptations, and adaptations are heritable features that improve an organism's ability to survive and reproduce. So, for example, some people produce more gametes than others, and that is an adaptive feature that helps people to pass on their genes, to reproduce and pass on their heritable traits to the next generation. And short-term changes in an individual does not constitute evolution, though. So evolution occurs over generations in a population. So I wanted to touch on this example as a natural selection thing, natural selection example. So antibiotics create natural selection for resistant bacteria. For instance, so if a bacteria, if a population of bacteria, um, so like exists for a certain amount of time, it mutates. There are some random mutations going on, and there's certain genetic diversity within the population of bacteria. And let's say out of a million of individual bacteria, there are 10 that are resistant to particular antibiotic. Okay. So if there is a subgroup of genetically diverse bacteria, a species, one species though, we're looking at only at one species, that is resistant to a common antibiotic, then if a person gets a bacterial infection with one species of bacteria, but that has high genetic diversity, then there is a chance that introduction of antibiotics will lead to the new antibiotic resistant strain of the bacteria, because the antibiotic will kill off all of the individuals that are susceptible to the antibiotic, but allow the individuals that are resistant to an antibiotic to survive and maybe thrive. But if there's no antibiotic, then bad news is that you will still have bacterial infection, but good news is that you will not develop a strain, which may lead to said outcome nevertheless. So the concept that I want you to take away here is that um, the the bacteria that are resistant to antibiotics kind of have to be present in the original bacterial population in order to, in order for the resistant strain to become more prevalent after the treatment with antibiotics. Okay. And it's quite a problem in hospitals these nowadays, the antibiotic resistant bacteria, because of basically people come to hospital when they're sick, and if they're sick with bacterial infections, the bacterial infections are treated with antibiotics. And because of the prevalence of the treatment of bacterial infections with antibiotic, there's a higher and higher probability of developing these antibiotic resistant strains. And in fact, there are new and new ways of treating bacterial infections that are emerging, and they deal with like cyclical treatments with antibiotics, and not like one treatment, but treatment like with lower doses, but for a longer period of time, to kind of balance out the the prevalence of the genotypes within bacterial populations, so that like there's less probability of developing the fully antibiotic resistant strain of bacteria. Uh, Questions about this example at all?
So we're at a pretty significant disadvantage because antibiotic or bacteria can mutate so quickly and we can.
Well, yeah, unfortunately.
Okay.
So, yeah, bacteria can mutate much more faster than our cells, because the reproduction rate of bacteria is, well, they reproduce like every second or so.
I mean,
But go ahead,
think how
Dylan.
simple compared to One word.
Oh, there's something wrong with your mic. It's like somebody's tapping on it. So
Hello.
yeah, it's better.
Okay.
Go ahead.
Oh my God, I'm drowning. I'll type in chat or something if I record.
All right. Yeah, so bacteria reproduce faster than us, and basically every time the DNA is replicated, there is a chance of mutation. So if this replication gave rise to a mutation that kind of caused the bacteria to be resistant to the antibiotics, then introduction of the antibiotics may result in the dominance of the strain of bacteria that is resistant to the antibiotics. But it's important to understand that in order for the antibiotic resistant bacteria to appear, there have to be some individuals in the original population. They cannot be developed because the antibiotic was introduced. They had to be present in the beginning before the antibiotic treatment started. Okay, this is from my experience. It's a common misconception that treatment with antibiotic kind of makes the bacteria to mutate. It does not. Bacteria mutate randomly, and the treatment with antibiotic just selects for the bacteria that already have this antibiotic resistant mutation in their genome. Okay, it's 10:21. I went a little bit over. So are there any last minute questions?